Okay, I will give you now a presentation about the scientific view, and I will intermingle this some work of ours with very general progress that has been made in the field over the last 20, 30 years. So in principle, if we talk about CRISPR-Cas, I always would say it's a revolution. It's not only an evolution, it's a revolution. And I want to tell you why in this presentation. So if we really talk about agriculture and the influence CRISPR-Cas will have on agriculture, we first have to think about what is plant breeding all about. And what you see here are in principle different monsters that breeders produced out of a plant. In the middle, you see a brassica plant, and this brassica plant looks indeed like a very normal plant. But if you then look at all these different monsters like broccoli and kale or Brussels sprouts, cabbage, it's really amazing what classical breeding could achieve. But the big problem is getting all these fancy plants. What was lost was genetic variability. So you could get out of brassica all these plants, but you, of course, cannot get any more out of kohlrabi or Brussels sprouts, now a broccoli. So in principle, a very extreme problem of plant breeding in general is that genetic variability is lost. And the question is, of course, how the hell can we get back to genetic variability? And there is one very brutal brute force method to do this, is to just induce damage into DNA. So if you induce damage into DNA, you create a situation that you change genetic information. This is, of course, a very destructive way to do it. And you might really think about why the hell would this be a benefit also to breeders? The thing is, you have to think about that even if you destroy information, you knock out genes. And if you have a gene that is in principle negative for a farmer, it might be really beneficial to do things like that. So I just want to give you an example. If you think about cereals in the wild, they have very long stems, very thin stems. And this is great for growing fast, for competition in the wild, but it's not really good if you have cereals in a field, because if a storm comes, the storm will in principle destroy this field very easily. And what would be much better would be to have short-stemmed cereals. And if you destroy a gene that is really responsible for these long stems, you will end up in short stems. And therefore, it makes a lot of sense to indeed just go into these plants and destroy genetic information, because you might get traits that are positive for agriculture. And in principle, this destructive technology is by far not new. And as you see also here, if you destroy genetic information, you induce double-strand breaks. These double-strand breaks have to be repaired. So this is exactly what we do even by now with CRISPR-Cas. But before, all these years ago, in the middle of the last century, when it was first discovered that such technologies work, this was done by X-rays. And with these X-rays, you, of course, induce hundreds and thousands of double-strand breaks at the same time. And this, in principle, works in plants because, in contrast to animals or to humans, if you put high doses of radiation to an organism, this plant cannot get cancer. So in principle, you are really able to induce a lot of mutations. Most of these mutations are bad mutations, but once in a while, you get a good mutation, like this short-strandness. And this, in principle, is a technology that was really developed on a level that you have to screen hundreds, thousands of plants, and you also have this negative by-effect that you, besides this mutations, which is interesting for you, you induce a lot of different other mutations, and you have to cross some of these mutations out because they are negative. But in the end, this technology was used efficiently for breeding since 17 years. And I don't know if you all realize or know that worldwide 3,000 different agronomically used cultivars were produced with this technology. And you benefit from this technology every day. So for example, maybe you had for breakfast already a grapefruit. This grapefruit was most probably seedless, and the seedlessness was produced by X-rays. Or another very prominent example is maybe you had yesterday evening a beer. All short-stemmed barley grown in Europe was made by this technology. So in principle, this is a very, very well-known used technology in breeding, and it's used all over the world. It's very efficient, and it's well accepted. Knowing this, it became clear since the middle of the end of the last century that one might be in a position to control this better. And if you think about inducing breaks in genetic information, you of course think about nature, you think about the fact, how is biology doing this? And obviously there are many situations in biology where genetic information has to be newly combined, for example, during inheritance, as shown here, where the chromosomes of mother or father are newly combined. And the first step is, of course, you have to cut this DNA at specific positions, or also switching of antibodies, for example, in humans. You also have to cut DNA first before you create the genetic information of a new antibody. In many other respects, these problems arise. And of course, nature developed specific kinds of enzymes for this. Enzymes then can cut DNA specifically. And it was clear from the start, if you get a handle on enzymes that are specific enough that you can induce only one or a very few breaks in the genome, we could really start to manipulate the genome. And the first step was, and this was just mentioned already in the introduction, at the beginning of the 90s of the last century. So we published our first publication in 1993, showing that there is an enzyme in yeast, IC1, that was specific enough. It has an 80-mer recognition site, as you see here, that was specific enough to be used in a plant that you only induce one break. And this gave us this unique opportunity to now, for the first time, understand what can we achieve, what different kinds of genomic changes can we make. I mean, there was this limitation that we can only induce this at this specific site, but still we could start to understand the whole process. And so we could show that in principle, if you induce a break, you will be able to knock out genes there. And this is the main pathway. It's called non-homologous end joining. So the two ends are brought together. There are in principle two sub-pathways, classical end joining, alternative end joining. It doesn't matter so much here. And this, for this audience, I don't go into details here. But the point is that if you induce a break and you have rejoining, very often mistakes arise. And so you can use this technology as a knockout for genes. Of course, as you see here, you cannot really predict exactly how this product will be made. This is different. You have a second kind of double-strand break repair. This is called homologous recombination. In homologous recombination, you offer the cell a specific matrix that has the same kind of sequence, at least at the ends, like it's shown here. This is tDNA, the black boxes. And then you can, in principle, if you induce a break in the genome, you can integrate at a specific position this DNA with this new genetic information. And because these homologies are copied faithfully, you can really induce a change that is specific for this site as you want to have it. We are really able this way to induce specific changes. So the trouble is a little bit that homologous recombination is not as efficient as non-homologous end joining. So you have to screen at least 100 times more events to get a homologous recombination event. In principle, this is important also if you see this different kind of changes for regulation, and you will hear more about regulation later on. What I show you here is how this was classified by a commission of the European Union in 2011. So if you have end joining, induce a break with end joining, and you induce a small change in the genome, which is always less than 20 nucleotides, you have a kind of event which they call SDN1. If you, in a way, change a few nucleotides by homologous recombination is called SDN2. But if you integrate a long sequence, a sequence that is more than hundreds of nucleotides, then you have SDN3. And this is in principle something very similar to what people see also if they integrate a transgene, there is then foreign DNA integrated. So SNS1 and SNA2 are very similar because they, and I will come to this later, are not being able to be really identified as foreign. Okay, so in principle, talking about history, another important point in this history was, of course, then to develop enzymes that can not only cut at one specific site, but can cut at different sites in the genome. And this is shown here. And what you see here is we started in '93 with homing endonucleases, and then people developed zinc finger nucleases. These are transcription factors that are able to recognize specific sequences combined with a restriction endonuclease to induce breaks. This didn't work very efficiently, and later on also transcription activator nucleases were developed. They worked much better. They had a transcription activator sequence of a bacterial pathogen, plant pathogen. And in principle, these technologies allowed us already before 2012 to really induce at specific breaks in the genome of plants. And we could, via using these enzymes, already achieve new traits. So this technology is by far not as new as you might think if we talk about the CRISPR-Cas revolution that started in 2012. But indeed, it started in 2012 because in 2012, Emmanuel Chapoutet and Jennifer Doudna could really show that in bacteria there's this enzyme Cas9, and this enzyme Cas9 is a protein combined with an RNA molecule, and this RNA molecule, which is called guide RNA molecule, is in principle able to direct in a very easy way sequences to specific positions in the genome, and the specific positions in the genome, are can be programmed by just using different guides. And the nice thing is, of course, if you have these guides here, as it's shown, you can not only use one guide, but you can also use several guides, and then you can induce several breaks at the same time. And you might ask now yourself, what was really new and why this revolution only came in 2012, and why did these two ladies get the Nobel Prize in 2020? Well, this molecular scissor was so much easier to handle than all the other scissors. A zinc finger, I remember you had to wait for half a year if you ordered one, and then you had to pay 20,000 euro. And with CRISPR-Cas, you just order primers overnight, and next day for 20 euros, you have a new enzyme. And on top of this, the Cas9 enzyme is cutting so much more efficient than the enzymes we had used before. And that's why we now see this fantastic, great revolution in biology. This was initiated by CRISPR-Cas. I know that not all of you are really experts in molecular biology, so I want to shortly also tell you where the hell is CRISPR-Cas coming from. This bacterial adaptive, it's coming from the bacterial adaptive immune system. So in principle, bacteria are infected by pathogens like plasmids or virus DNA, and then in their genome, they really have a memory system, a cache, where they just store part of this genetic information of these viruses, bacteria, and these genetic information is stored in clustered regularly short palindromic repeats. That's also where the name CRISPR is coming from, which you see here. And they really have some cache for many different infections. And what's now going on is the following, that these DNA sequences can be transcribed into RNA, and these RNA sequences are then processed, and out of this comes these guides, which I showed you before, and with the Cas9 protein in combination with the guides, you have a specific nuclease for the specific phage. And if the phage, for example, or the virus is again infecting the cells, and the enzyme is around that is recognizing a specific part of the nucleic acid of the phage. And then in principle, what happens is that the genetic information of the phage is cut, and the bacteria became resistant. So this is a system where you find this in many bacteria. About half of all bacteria have such a system. We started to use the system in plants. Many groups started at the same time. Some groups established it in rice, for example. We established it in Arabidopsis. Arabidopsis, most of you might know, is the model plant for plants, for plant scientists. So it's in principle a weed, but nevertheless, it was very important to establish the technology in plants, and we were able to set this up. And it's, of course, important not only to induce mutations in plant cells, but to bring these mutations to the next generations, which you see here, where you have a stable mutant. Sorry, this is in German. And this is in principle the first mutation we achieved in Arabidopsis. We knocked out a gene that is responsible for the brown color of the seed coat, which you see here. We cut the site, a single nucleotide was inserted, this red one. And what you see here, in principle, are the yellowish seeds. So the technology worked well in plants. And what we now and many other groups are trying to do over the last years is really to improve also the molecular scissors. So in principle, there's not only Cas9 or one specific Cas9 of Pyogenes, which was originally used by Gluten and Chapoutier, but there are many other enzymes. I told you bacteria have CRISPR-Cas systems all over, and so a very prominent system. This is also used now in the Cas in the field is the Cas12a system, which is also shown here in this slide. And a way to improve the things is that you adopt these proteins for plant use. And as many of these bacterial proteins are human pathogens that grow well at 37 degrees, it's of course unfortunate if you use them at 22 degrees to mutate plants. And therefore, we just to give you an example of some current research we did, we developed a Cas12a enzyme. This is much better in cutting than the classical enzymes that you find in this bacteria. What you see here, the green bars on the left side, the efficiencies is shown of our mutated enzyme in comparison to the wild type, which is shown in blue. And you see we really could enhance efficiency much with this approach, so that we are getting up to 100% cutting efficiencies. And this also means, of course, you have to transform much less plants than before, and it's much easier to get molecular changes, or also to get changes that are not occurring so often. And this is also shown on the right side. This is gene targeting homologous recombination comparison in blue of the newly developed enzyme by us with the enzyme that we found in the bacteria, or other people found the bacteria before. And you see also here the efficiency is much better. Now we are in certain cases at least to 7 to 8% of homologous recombination, which is much better than at the time when we started. So in principle, there is a lot of technical developments. I don't cover all of them. There are also base editors by now, where you can change single bases in a way to induce point mutations. But what is very important is, of course, now to apply this technology to plants, and also to give an evaluation about the risks of this technology. And you always have to keep in mind that if you induce a break, it's an artificial way that you start the reaction, but in the end, it will be a natural way how you repair the break. And so DNA repair is a natural process, and if there's double-strand breaks occur all the time in a cell, so in principle, in the end, you cannot discriminate whether a break was induced by X-rays, a break was induced by a molecular scissor, or it just happened in nature. We also have a lot of experience with molecular scissors right now, over 30 years. Better and better molecular scissors are developed, especially for medicine, because you have to be very careful to induce only one single double-strand break, to avoid cancer, for example, also in patients. The nice thing is, as I told you before, this CRISPR-Cas is very quick, it's very precise. As I said, we have hardly off targets. And in the end, these plants, and there is no way how we can see this different, and this will also be important, of course, for regulation. Spontaneous mutations cannot discriminated from CRISPR-Cas induced mutations. And I just want to give you now an example to make it clear to everybody why I also in discussions with politicians always say we are by now better than nature with this technology. If you think about what I told you before in the beginning, it was about classical mutagenesis. Classical mutagenesis induces thousands of mutations. For example, in barley, there is an estimation that you have at least 10,000 secondary mutations besides the mutations you have. And all these 10,000 mutations, of course, you bring out in the field. And if you think about this, and if you also think about the fact that if you go to a barley field and just take two sister plants and determine the sequence, you would also find differences. So from the mutation rate alone, you would find 100 differences, 100 nucleotide mutation differences between two plants in the field, two natural plants in the field. And then we come with our molecular scissors and we induce one single break. Maybe there is a second break offsite or third offsite, but this is below the noise of mutation. So that's why I think we are really now getting better than nature with this technology. And therefore, it's also completely, nobody will be able really to discriminate a plant that has 100 natural mutations and a CRISPR-Cas mutation from a plant that just has about 100 natural mutations. Besides this safety considerations, I think, of course, it's in such a talk, of course, important also to show you a few examples what we can really achieve with this technology. One important aspect is, of course, pathogen resistance. If you have pests, you often fight these pests with chemicals, with pesticides, and you all know that there's a lot of critique on the use of pesticides in agriculture. So a good way to reduce reduce pesticides, to use less pesticides, is to develop plants that are resistant to CRISPR-Cas, if they are resistant to fungi or bacteria by CRISPR-Cas. Just one example, the first that has been developed some years ago, this was a resistance against powdery mildew infection in weeds. This is a fungus that really ruins the fields, as you can see here. And in principle, there's a very simple way to induce resistances. And this is due to the fact that there's always a specific interaction between host proteins and bacterial proteins. And if you just take out a host protein that's specific for the infection process, these plants cannot be infected anymore. And this has been done also in wheat by knocking out the MLO gene, which is important for this infection, for this interaction of the respective fungi with the plant. And indeed, a group in China was able to knock out by molecular scissors all these three genes. And this was work done by Shaisha Gao. Shaisha Gao, a good friend of mine, is really the leading scientist by now in the CRISPR-Cas field in plants. And she could really show that if you knock out all three copies of the gene, you might know that wheat has three different genomes. So if you knock out all three genes in the three different genomes, you get these plants that don't show the symptoms anymore. This is on the right side of this picture. And in principle, she could show that CRISPR-Cas can really lead to resistances. And you can apply this not only, of course, to wheat. Scientists also applied it to tomato and also to grape, for example, recently. So this is really a big step forward. And there are other very important problems that we face at the moment. I don't know whether you know that the classical banana, the Cavendish banana, there's a major threat that our kids might not eat these bananas anymore because there's also a fungi that is hard to combat. And scientists are working hard at the moment with CRISPR-Cas to develop bananas that are resistant to this fungus. Besides these examples for improving resistances, of course, a very important aspect is also to improve the product, to make crops better for human health. And one example that is already used in the field is gene-edited soybean. There is a company, Calixis, in Minnesota that developed this, and the soybean has the soybean oil has in principle more healthy fatty acids, and this might also in the long run help in principle also consumers to stay more healthy. For the industry, it's interesting to manipulate starch. You know, you have branch starch and you have normal linear starch, and the more branch starch you have, the better is, for example, to make for industry to make gels or make yogurts or whatsoever. And there have been a lot of approaches by now in different plants, in corn, in potato, and as you see here also in cassava, to obtain starch that is branched, and that is very interesting also for industry use. And last but not least, I think a very, very important application where people already achieved a lot of progress is to produce gluten-safe wheat or other gluten-safe cereals. As you might know, there is a celiac disease where people have problems if they eat cereals because of gluten, and they have very strong reactions to this. And of course, it would be a strong relief to this all these people, which are, as far as I remember, is about 1% of the population, if you could produce cereals and they could have a normal life eating those cereals. Besides this knockout of genes, another application I just want to show you, and this is an application developed by Zach Lippman, also a good friend of mine working at Cold Spring Harbor, is to manipulate the expression of genes. So in principle, if you think about plant development, there are master switches in development, how fruits develop, if fruits are bigger, if fruits are smaller, if more fruits are produced on a tomato. And for this, it doesn't make sense to knock out this master switch, because then you wouldn't see anything, no fruits at all, but to manipulate its expression. And for this, he really mutated the promoter regions of these master switches, which resulted in less or more expression. And as you see here, he can really engineer a trait variation like it's shown here, how bigger and smaller tomatoes using this technology. So we are really have now a lot of tools in our hand to make better fruits, to make fruits that are really interesting for the consumer, but also are interesting for the farmer. But of course, we won't stop here. So till now, what's coming now to the market are single traits, and you will hear more about this later on. But as a scientist, I also want to give you a vision, of course, and the next step, what I always call, is breeding at the speed of light. I mean, over the last 20, 50 years, we accumulated a lot of knowledge about different genomes. We have gene banks where we have a lot of different wild species, which in principle have very attractive resistances. The problem is a little bit to get all these resistances into our cultivated plants where there is no genetic variability anymore. And part of the reason is, of course, that you have this genetic information organized in chromosomes, and half of the chromosomes where there's a lot of genetic information you cannot use for breeding because there is no genetic exchange possible in this region. So it's very hard really to get traits from a wild species to a cultivated species. And there are two very interesting developments recently that will really revolutionize breeding. This domestication by multiplexing and chromosome engineering. Domestication is most probably the most promising progress that has been achieved since CRISPR-Cas has been used in plants. and I want to come to this first. So in principle, my good friend Dan Wouters, we share our enthusiasm for nucleases, site-specific induction of breaks since I think at least 25 years, was able to get a wild tomato plant, to transform a wild tomato plant into a cultivated tomato plant. And how did he do this? Well, in principle, we know exactly, because of all the information we accumulated over time, what kind of genes have been changed during breeding to make a wild plant into a to bring a wild plant into a cultivated plant, like fruit size, to get rid of bitter taste and so on and so forth. And what he did is now he used this multiplexing function of Cas9, inducing several changes at the same time in these plants, and then he could get in more or less one generation all these changes at the same time, and he ended up with a domesticated tomato. And this is really amazing. And of course, you can also apply this to other plants. Just 2 or 3 weeks ago, a paper was published in Cell, as you see, where a wild rice species was transformed by a row of Chinese groups into a cultivated rice. And this wild rice has, of course, other kinds of resistances. And that's why it's very important to get these resistances now in a cultivated plant. And another example I want to tell you is ground cherry. Here you see also the first results of domestication of ground cherry. On the left side, you see the original fruits. On the right side, you use the bigger the engineered fruits. And in principle, this also gives us the opportunity to think about whether it would not be possible to really take a lot of species, wild species that are, for example, heat and salt resistant, for example, also species in Africa like teff, and really develop out of these species new croplands. There's always this big problem in agriculture. How do we react to the global warming? And of course, we need new kinds of crops, but because we have all the trouble with genetic variability, with the highly developed crops we have now, it might be much harder to get heat and salt stresses in these crops than to really take wild species and make new crops out of them. I think this is really a promise for the future. And this is something that we all have to take into account when we talk about the CRISPR-Cas technology. I'm coming almost to the end. I want to tell you a little bit about chromosome engineering, because this is also, I think, an important new tool to solve problems in breeding. And of course, I also tell you this because we developed this recently. In principle, one of the major challenges of breeding is to break or change genetic linkages. And this is especially important if you have two genes like shown here on the right side that are close together on a chromosome. So in principle, if this is a positive trait like a resistance and then negative trait like a better bitter taste for a fruit, you never can get rid of one or the other because they are too close for classical inheritance crossovers. And therefore we thought maybe we can do this by cutting, making a cut between the two genes, and then at the same time cutting another chromosome and exchange chromosome arms. And with this exchange of chromosome arms, then you really can segregate the two traits, the positive trait and the negative trait. And indeed, as I show you in this slide, we were able to do this. And what you see here in this picture, these are the original chromosomes, Arabidopsis chromosomes, the blue one and the red one. And here we induce this change, and this change now we have a new chimeric chromosome that nature has never seen before. And this indicates that we can really use this technology for breaking or creating new genetic traits. And another interesting problem in breeding turned out to be inversions. So only recently, because of this modern sequence technology, long reads, people realized that in many cereals, you get a lot of you get a lot of inversions that arose in nature. So inversions apparently occur very regularly, but all these regions that are inverted, like it's shown here, you don't have any genetic exchange. So if there are genes together on this part of the chromosomes, you never can get them apart. And of course, it would be clever to then just change the linkage in a way that you revert the inversion. And then you could get genetic exchange. And you could also, of course, do this the other way around. If there are several genes that you don't want to segregate away from each other, don't want to let you segregate away from each other because they are good traits, you can do also the opposite. So in principle, we also set out to set this up in plants. And what you see here is in principle the result. Here we have a red and a green, and we exchange now the chromosomes. This was also done in Arabidopsis. And this is just this is a chromosome mark in red. And here you see the one orientation, red, green, and here you see the other orientation, green, red. And so we were really able to also inverse a megabase region in Arabidopsis chromosome. And this, of course, was recently also shown by Corteva scientists to work in maize. And this is not the end of the story, because the end would be, of course, to show that we can also get back recombination into a region where there was no recombination, in this case, in the last 5,000 years. And indeed, as I show you here in this slide, the blue line shows you the recombination rate in this inversed orientation, and the orange one in the normal orientation, and we could get back also recombination. So we can reactivate recombination in that regions, and we think this will also help a lot breeding in futures, that we can get the best trait combined in cultivated plant species. So the take-home message is very clear from the scientific point of view, and I just sum this up in one single slide. CRISPR-Cas is a key technology that will enable us to set up sustainable agriculture for the 21st century, and we should really, as a scientist, I have to say, is also use this technology in Europe, because otherwise it's really, you will not be able to face the challenges. Of course, CRISPR-Cas is not the only key technology for sustainable agriculture, but it's a very important technology. And with this, I'm at the end of my talk, and here you see my group, and I want to thank not only also all my group members, but especially the European Research Council for funding us over a very long time period and making it possible that we, although we are in Europe, that we kept pace with the fields that is mainly in China and in the US, where also important developments are. And with this, I'm happy to answer your questions.